Okay, so chapter 6, we're going to continue kind of talking about chromosomes, but we're going to talk in chapter 6 about chromosomal imbalances, chromosomal structure, imbalances in the number of chromosomes and the karyotypes and things like that. As I told you before in chapter 4, there were problems, right? But do you think those problems are probably, once you start doing them, they're much more manageable? They're just kind of an extension of the problems that we did in chapter 5. They're an extension of the problems we did in chapter 4. But having said that, after going through that second exam, it's a really good idea to start working through those right now and getting really comfortable with them and going through. And usually students do that after taking that second exam. And the result of that, most of the time, is they score a lot better on the third exam. So I'm encouraging you, go through, do them. Take those quizzes at the end of the chapter and start doing those problems. And I think, did I post the chapter 5 problems? I don't think, did I already post chapter 5? Okay, I'll post chapter 6 too, so that you can kind of go start doing these. We're going to run into a different set of problems in chapter 6, looking at, and in their own right, they're a little bit, it takes a little while to figure out how to do them. Once you do, just like those other problems, they're not so hard after you do them a few times. Okay. So, the way that the chapter is broken up or outlined, can you see now? We're going to talk just a little bit briefly about cytological techniques. These are just basically the techniques the scientists use to figure out and look at different chromosomes, to compare different chromosomes, to basically look at the geography of a chromosome. Christian?
So which chapters will be on the next test? 5, 6?
Yeah, so apparently we got through chapter 4 on the last one. So on the next test, it's going to be 5 through wherever we get through.
Okay.
So depending on how fast we go or not. Maya?
Is the test still going to be what it's scheduled to be on the
Yeah, I always keep this. When is it scheduled for?
It's next week.
Oh wow, so there might not be that much on this exam. Yeah, no. So I never switch the dates. I just adjust the material. So it'll probably be 5, 6, and 7. I would imagine. I bet that's what we're going to get through. Next, what, Thursday? Wow, that's a weird scheduling. Yeah,
Because we have the final on the 28th, right?
yeah. So then the final there'll be more stuff on that final. Wow. So well, that's it's good for you all because they're worth all the same, right? So this is gonna be the one if you really want to bump your grade. These 3 chapters, the problems aren't going to be as hard, but you'll see that it's still. That's not so bad because there's still problems enough in these 3 chapters that you'll wanna. It'll still be a good exam. Don't worry, you'll still get your money's worth on exam 3. Don't fear.
One other question about the final. So just to clarify, the final isn't comprehensive?
Yeah, the final is not comprehensive. There's enough overlapping in these chapters that I don't do a comprehensive final. So yeah, so it will be from wherever we get on chapter 3 or whatever we get on exam 3 to the end of the semester, and that's what the final will be on. So it'll be a lot on the molecular stuff probably. Maybe some population genetics, maybe some evolutionary genetics. We'll have the transcription translation as a flipped lecture, so that they'll have. That's before the next exam, right?
No, it's right after the next.
Oh, right after. Okay, yeah. So there'll be a little bit more stuff on that final, but it's not comprehensive, so it should still be manageable. Okay, so then we'll talk about polyploidy and aneuploidy. These are different numbers, different sets. These are basically these unbalances or not the sort of the normal situation when we're looking at chromosomes. And then at the end, we'll talk about chromosomal structure and rearrangements, how chromosomes will flip around and change conformations and things like that. Okay, so that's basically what we're going to be talking about. So cytological techniques, scientists use these techniques to stain the chromosomes. Initially, they did it just kind of staining it just so that they could see those chromosomes under the microscope. We then developed other techniques, and we'll just talk about, just briefly, how they would differentially stain a single chromosome and create these bands and things that we could then get a better idea of what's going on in different parts of chromosomes, and eventually we want to identify the geography of a chromosome because we want to figure out where the genes are that code for different things and be able to link that to a place on the chromosome. Now, Christian, you're not trying to ask a lot of questions, so we don't get through much stuff, are you?
So the staining, I've always been curious to see just logically how you get one chromosome stained and nothing else in the cell. I mean, it just seems like hard for me to believe that you can get a needle that's small enough that you can get.
Well, usually what happens, so
How does that
which
happen?
which staining are you talking about? That it does just a single chromosome?
Well, and actually, I guess just any staining really, like
So the stain, if you remember, so this is all called cytogenetics. Let's go to the next slide. When we do the stainings, what we do, if you remember, what is a chromosome made up of? Proteins, right? Histone proteins and DNA and things like that. And so usually in the beginning we would just stain them, and the things that would stain were like the cell membrane, the chromosomes. The name chromosomes comes from the fact that they had the color, they took up the color. So in that case, all of the chromosomes, like when we were doing the onion root lab, right? All of the chromosomes got stained. We then, what we'll see is we then developed some techniques where the stain was taken up differentially within a chromosome. So some parts of the chromosome would take up a whole bunch of the stain. Some sections of the chromosome would take up just a little bit of the stain, and that was just based on mostly how tightly compacted the histones were to the DNA. Or eventually, we developed staining techniques where we could attach specific fluorescent proteins to specific parts, or not fluorescent proteins, but fluorescent dyes to specific DNA sequences. And that Okay, so we're just going to go through and look at just briefly talk about a couple of different banding techniques. The quinacrine or quinacrine banding helps us to distinguish the different chromosomes. So here's a bunch of chromosomes that have this different banding. What you do with quinacrine banding is expose it to that, or you expose it to the dye, and then you expose the quinacrine molecules to UV, and they'll fluoresce. And so here you can see that different chromosomes are fluorescing differently. So Christian, here, it's not that it's just necessarily one chromosome or the other, but different chromosomes are going to fluoresce at different parts depending on the composition of the DNA and histone proteins and things like that.
So you just like inject the dye
Into the cells, yeah.
cell and the chromosomes uptake the
Yeah,
dye.
yeah. Well, and you don't have to inject it. You can extract the cells, put them on a petri dish, kind of, or however, like how we did it, and then just expose the whole cell to the dye. It goes into the cell, selectively goes and concentrates in these different areas, expose those cells and the chromosomes to UV, and it'll fluoresce like that. And that's the general sort of protocol of all chromosome staining techniques.
Those posters that have like all the human chromosomes, maybe you've seen those before where they lay them out and
Yeah.
they're all colored. Is that just
Yeah, we'll get to those here in a second. Yeah, so, and there's actually, sometimes we do a lab, a cytogenetics lab, where we read a paper where we have that, we look at those, there's a figure in that paper. I now have it as a makeup lab, so some of you, has anybody done a makeup lab yet? Okay. So, I use it as a makeup lab, and there's a whole bunch of questions and stuff. You would see that, and so, and
digitizing in that? Do
we'll
they digitally enhance those? Is that kind of how they do that?
I don't know, not necessarily, I don't think. So here, and that's a banding that we'll look at here in a second. That's it. And in all honesty, there's other kinds of bandings that we're not going to talk about. I'm sure there's types of stainings that I don't even know about. So there's a lot of different things, and I'm just trying to give you sort of a sample of some of the different kinds of staining. Here's the Geinsa stainings. These are ones that I think you do in some other labs here. I can't remember which class it was, but I know students have done these before. These are sort of a traditional ones. They're non-fluorescent staining, but again, you see that different parts of the chromosomes are taking up the stains in different amounts. That allows you to, one, distinguish different kinds of chromosomes, right? And two, sometimes, and we'll get to this in a little bit, distinguish between parts of a chromosome. Okay, so here's one that's called chromosomal painting. Chromosomal painting, in chromosomal painting you develop probes based on DNA sequences where they have fluorescent dyes attached to them. We'll talk about fluorescent dyes too when we talk about sequencing. It's kind of a similar thing going on here. But basically you can take and develop different not markers, but different probes that have a specific sequence of DNA that will then go and attach to a specific sequence of DNA and then fluoresce there. And so this is one that we developed probes based on human DNA and then exposed that these are given chromosomes. And so we say, okay, in humans, we have some sort of, just pick any kind of gene, and we're wondering, okay, do gibbons express that same kind or vice versa? We look at a chimpanzee probe and say, okay, is the, not cytochrome oxidase, is the MYH2 gene in the chimpanzees being expressed and doing the same thing in humans and vice versa? And then you can go through and you can see, okay, here are these probes. One, you're going to now identify that this is this homologous pair of chromosomes, right? But not only that, now that we're doing this with specific sequence data, we can also look, and especially if we develop those probes based on a known gene, we can say, okay, what's going on? How does the number 2 chromosome in humans compared to the number 2 chromosome in chimpanzees, right? Do they have the same kind of genes on them? Do they not? And we can use this for a lot of extra things. These are some of the added features of being able to not only distinguish the chromosomes with these different kind of techniques, but also look at homologous regions of chromosomes, and then begin to reconstruct the chromosomal sort of history, like what happens. And we'll get, I think I still have a link to a little video here from the video that you saw in 1110 maybe, the one about Dover trial. Does anybody remember seeing the Dover trial one, the 2-hour lab?
That this semester.
Yeah, they just did it, yeah. So there's a part of that that has to do with this same sort of thing. So here's a human karyotype with chromosomal painting. We can use these to identify the different chromosomes. We can also use those sometimes. So say you were looking at an individual that you suspected had Down syndrome, you could then be certain that it was trisomy 21 because you can identify that specific chromosome. Okay. We use these before we could use these staining techniques. We could only really identify, if you look again, the nomenclature of chromosomes. The largest ones we call number 1. Then we go down from there. Before we could actually stain the chromosomes, we could only really categorize or look at human chromosomes and identify major groups. So like this was a major group. They were too similar in size to really distinguish between them. It wasn't until we were able to begin to have these more advanced staining techniques that we actually could identify each of the human. Okay, so here, the next step then is to stain them, but then to begin to describe the geography of a chromosome and name these different bands. So the higher the resolution of whatever technique that we're using that stains these bands, we can then begin to look and identify certain regions. So here, this is a chromosome. Did I say what? Oh yeah, chromosome number 5. This is what's called an ideogram. So this banding pattern in the chromosome, through these staining techniques, we're able to generate these ideograms or ideograms, and we can begin to talk and communicate about different things that are going on in the different chromosomes. In order to communicate, we have to name the regions. We call the short arm P for petit, French for petit. The long arm Q, I'm not sure what the Q is for, but you can when you're talking to another scientist, you can say, okay, at P 13.3, and you know exactly the region that you're talking about. You can say Q 21.1, you know exactly the band that we're talking about. We'll look at how that can be important later on when we begin to identify mutations and deletions and insertions and things like that. Okay, so this is an ideogram with a pattern. This is the nomenclature for identifying different regions within the chromosome. Okay, so just a couple of vocabulary. I just want to explicitly give you a list of vocabulary. Some of these things we've already talked about a little bit. When you hear ploidy, ploidy just means fold or set. So polyploidy means many sets of chromosomes. Euploidy is the organism's U, or true, right, or normal, quote unquote, right. The euploid organism has its complete set of chromosomes. So in humans, the euploid is a diploid. We have 2 copies of each chromosome, right? A triploid then means that you have an extra set of chromosomes, right? So if a human was triploid, if we had a triploid human individual, how many chromosomes would they have? 3, or how many total number? 3 copies of each. Therefore, you would have a haploid number. So the N, 3N, the haploid number is 23, right? So you would have 69 chromosomes. Do we know individuals with that? No, because we can't adapt to it very well. We see, though, that this can happen fairly readily in plants and some other organisms. Okay, the triploid condition. Tetraploid, 4N, etc., etc. Anything that has more than 2 copies of each gene. And you guys are doing a lot of fist bumping back there today. You guys are excited. I've never had anybody fist bump over polyploidy. That's good. It's good to see you guys are like excited. Okay, so anytime there's over 2N, right, or anytime it's more than the diploid number, we call that polyploidy. Okay, so there's a whole bunch of copies, right? Aneuploidy means that the organism has particular chromosomes or part of chromosomes that are under or overrepresented. This is the sort of unbalanced part. Aneuploidy implies a balance. That's when we have situations and conditions where of sterility and things like that. And we'll talk about all of these, right? And then lastly, rearrangements that we're going to talk about at the end of this section is when we have changes in the structure or even changes in how the DNA is arranged within the chromosome. And we'll talk more about that. Okay, so here are all the key points. Staining, what we use staining for, and how that tells us about. Okay, so polyploidy. Extra sets of chromosomes of an individual. It obviously can affect an organism's appearance and specifically fertility. When we talk about polyploidy, the vast majority, do you have any botany majors? No? The vast what's that? Oh, you're dual, right?
No.
Where's Carl?
Carl's
Oh, yeah. Shoot, we're going to talk about plants today and Carl's not here. Oh, maybe we won't talk about plants. We'll save it for next time.
Save it for Carl.
Yeah, we'll save it for Carl. What's that?
Is that a white street from, or what is it?
I don't know specifically.
My sister just got from.
It's spotted like that? So anyway, so polyploidy is usually we think about plants. Plants can readily adapt much better than animals to extra sets of chromosomes and things like that. But that doesn't mean that we don't see polyploidy in animals. There's fish that are polyploids. There are frogs that are actually polyploids. I'm not sure about what the frog species are that are polyploids. I think this is just a frog picture, generic picture, yeah, off of the internet. And fish. And so we do actually have polyploid individuals in animals, but it's much, much more rare occurrence. Polyploidy in plants, if we look at the plants that are polyploidy and we start listing off different ones that are, we see that a lot of our agricultural crops are actually polyploidy. So polyploidy can confer differences in appearance too, and that's important for human agriculture because a lot of times that gives us better crops. So here's bananas. Bananas are polyploids here, or daffodils or something. They're polyploids. If we look at all plants, half of all plant genera have polyploids. If we look at grasses, 2/3 of all grasses, just species, are polyploids. So polyploidy occurs a lot in plants, and you can sort of think about it for a second, and it's pretty, it's not a big surprise because what happens when you're polyploid? If you can deal with the imbalances of the chromosome or of the genes and stuff like that, what does having polyploidy do for the organism? Big adaptation, right? You have more genes, right? And so it gives you a whole bunch of more material to work with from an evolutionary perspective. Okay. Other polyploids, strawberries, cotton's a tetraploid, strawberries an octoploid. One general effect of polyploidy is an increased cell size. For some reason, that also translates into more robust plants, more robust sort of healthy plants, and that's why we see a lot of polyploids in our food crops. They're larger and tend to be more robust.
That's great. So like all the, not all, but like strawberries, since they're super large, and like what's a great feature is because they're polyploids.
Yeah, and if you think about artificial selection, a long time ago with corn or with whatever else, as people began to artificially select different crops, you can see that they didn't know it, but probably they were selecting for plants that had gone through and had become polyploidy. And then each time they're selecting for that, they're selecting for the characteristics, but indirectly they're selecting for polyploids. And so we have lots and lots of plants. Polyploid. How come polyploids are sterile? Here, this is just an example of a triploid. You're all experts on mitosis and meiosis, right? it's not hard for us to think about what's happening with the chromosomes during meiosis to think that, oh, okay, I can see where it's polyploid. So here in metaphase 1, you are lining up with your homologous chromosome, right? If you have 3 of them, so we're just looking at one chromosome, if you have 3 of them, it's, might mess up how they're getting lined up. They could line up like this, up here in A. If they lined up like this, the upper 2 go to this side, the 1 goes to this side. The eventual gametes from this upper one are going to have an overrepresentation of that chromosome. The gametes from this lower one are going to have the normal representation, right, in the gamete. They're just going to have one, whatever chromosome this is. If sometimes they can line up like this, it can be really sort of messed up. All 3 of them can go to one side, none of them can go to the other side. In this case, how many normal gametes are you going to have out of the being developed or produced from this one? Elizabeth's shaking her head. None, right? You're going to have either a 0, an underrepresentation, or a massive overrepresentation. When you have these over and under representation, that oftentimes messes up your embryo and your embryo spontaneous. Pause that. Okay, so then if we can see why we get sterile individuals resulting when they have polyploidy, we have to answer the flip side of it. How do we get fertile individuals from polyploidy? When we went through, we looked at all these plants, strawberries, etc., we see that they are the result of polyploidy. They're a result of complete genome duplication, right? So how do those type of individuals form? What has to happen? This is a figure from the book that is just this general description of what is going on. Here you have 2 diploid plants, right? And it has this AA and BB. That's not a genotype. When you first look at that, you're thinking, okay, is this some sort of genotype? Basically, what each of these is representing is this A is representing the haploid component of that genome. And so you have 2 haploid components of the genome, it makes a diploid individual, right? 2 haploid components of the genome makes a diploid individual. So I've just threw some numbers in here. These numbers aren't in the book, right? But I think it's helpful to look at some numbers to kind of understand what's going on. So this diploid individual, big A, not the genotype, right, goes through meiosis. In the gamete, you have a haploid representation, right? You have 1 copy of each of those chromosomes. If the diploid number for this individual is 30, the haploid number here would then be what? 15, right? Here, the same thing with this second plant, BB, haploid number would be 14. But let's say, as we often see in plants, they actually can fertilize each other, they can mate, and they can have offspring. So here's your hybrid individual. What then is the diploid number for that individual? It's going to be 15 plus 14, 29. Now we have that imbalance, right? When your 29 chromosomes go to line up with each other, something's going to be askew. That's where the sterility comes from. That's what we're talking about in the previous slide. Do we see this? Do we see sterile hybrids in nature? Yeah, we do. And the most common example, usually when I ask people what an example is, what do you say?
Mule.
Right. So here's a mule or a hinny. So a mule is where mating between a donkey and a horse, where the horse is a female. A hinny is a mating between the donkey and a horse when the female is the donkey, right? So probably for physical stature reasons, we see more mules than hinnies, right? Because the female is the horse. If you look at mules and hinnies, they have a diploid number of 63. You're going to, reverse back 2 slides, and you're going to see there's where we have the problems during meiosis. So here, horses have a diploid number of 64. Donkeys have a diploid number of 62. You have that sterility. But in plants, plants and other organisms, like when we talked about the frog, we talked about the fish, where we do have polyploidy existing, something has to occur for us to overcome that sterility in the hybrid. Christian, question?
Since we've got the diploid number of 29 on that slide and 63 here, due to that imbalance, would that be considered aneuploidy?
Yeah, you could call it aneuploidy. Yes, that's sort of a semantics, but yeah, there is some sort of imbalance in there. In a more strict sense, you still have the imbalance in that case is how the genes are housed in the different chromosomes. rather than the number of copies of the genes. And so there's a slight difference between that. You could consider it, but usually we're talking about different numbers of genes that then have these physiological effects from that when we're talking about aneuploidy. But that's a good question. So here, what happens? How do we get, what is the general process that goes on so that we can go from being a sterile hybrid due to polyploidy to a fertile individual? Okay, so here again, we have this hybrid, it's sterile because of the meiosis is irregular, right? There is an event, this is the most crucial event that happens to balance that out. So let's say we go back to where we have 29, right? We have 29 as the diploid number. If you go through, and let's say you're going through meiosis, and what happens in meiosis, your chromosomes line up, or they get split apart. They go to, one part of the sister chromatid to one side, one part of the other side, or even let's say meiosis, where the homologous chromosomes line up. What would happen if all of the chromosomes go to one side and none of the chromosomes go to the other side? You would have this chromosomal doubling. What's the end result of a chromosomal doubling? You then get an even number. So now, going from 29, your chromosomes double, right? That then becomes what? 58, right? Now your new diploid, well, it's really not a diploid now. Now we're talking about a tetraploid, like we were talking about before, right? Your new tetraploid number is 58, but the important part here, total number 58, the important part now is that you can have even pairing between, let's say, in this, let's say that the plant, we'll just say, in the AA plant, you had a number 1 chromosome and another number 1 chromosome. Now these will be pairing up with each other, right? It might be very similar, so that's probably going to foster some tight pairing. But the point is that now you have an even number when you go through, when this fertile tetraploid produces gametes, the new haploid number is 29, the same number of chromosomes that the sterile hybrid had. And so the chromosomal doubling is, an important and crucial part of having fertile offspring from polyploidy. So, thinking about what we know, yes, this is a zoology class. Who's to do? Oh, where's Carl again? We need our botany major. Why is it that we see lots of polyploids in plants? What is it about plants? Self-fertilization is one, so that would be a good thing. What else? What can you do in a lot of plants? Are they able to reproduce by grafting? Like, have you ever grafted a plant? So they have lots of their cells are capable of going through, housing that information, going through and self-fertilizing, or just growing into a new organism. And so their sexual tissue is distributed throughout the plant. A lot of times it's not just in certain areas like we see in mammals. So there's a lot of things about plants that lend themselves to this process occurring much more frequently in plants, and that's exactly why we see it much more frequently. That's why we see lots of in plants. Okay, so for instance, one of the plants we didn't talk about, wheat. Wheat is a hexaploid. It has a 6N value. Here, going kind of just continuing on with that figure. Here we have 2 organisms, right? Remember, the A is the haploid number. This is 7. So the diploid number of this plant is 14. The diploid number of this other plant is 14. They form a hybrid, right? So they have a hybrid formation from that. The chromosomes then double. This is sort of how we are hypothesizing that wheat plants formed, right? And so we had some ancestors. They were both some ancestor in the past of the wheat. They both had diploid numbers of 14, but they were probably different species of plants. They hybridize. They form a hybrid here. The hybrid goes through a chromosomal doubling, right? The new, if this self-fertilizes, the new organism has a diploid number of what? 28, right? Good. So the new organism has a diploid number of 28. We then have another individual coming in here. The haploid number is 14. The haploid number of this new plant, new species of plant, is 7, right? You have form a hybrid here. The new haploid number is 21, so then diploid number is 42. When it goes through that chromosomal doubling, and then you have wheat. What we have is wheat. So why do we think that? Like, we have wheat now, right? How are we sort of coming up with this evolutionary story? You know, because couldn't you say, well, how do you know these are, you know, why are you assuming all this stuff? We just know that wheat right now has 42 chromosomes, right? Haploid number of 21. What sort of signal or pattern, when you start looking at the genes and things in wheat, should you see? What do you think? Corey doesn't know. Maya doesn't know. Anybody else shaking their head no? What would you think? If you were to look at, let's say, some, you know, protein producing gene in the wheat, that's a common gene, protein producing gene, how many copies of it would you see in modern wheat? Or how many copies, even maybe not functional copies, but sequences of DNA that are very, very similar? How many would you see? Corey, did you raise your hand? I thought you said this. Did you do that? No? I think Corey put up 6 fingers, and he's right? Because if these species are hybridizing with each other, they're probably fairly closely related. They probably have very similar genes, and you would expect to find a copy of the gene here, 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 and here. Should get 6 copies. So that's where we're inferring these. That's where we sort of go when we infer these evolutionary stories. This is the best explanation of the evolutionary story. So it's a good story, right? Is it proof that that's exactly what happened? Maybe, maybe not. Who knows? Is there a way to test this? How could you test it? Yeah, you could maybe try to, you know, replay that evolutionary history in a lab and go through and force, you know, hybridizations, treat them in certain ways that would facilitate chromosomal doubling, and then recreate history. And that's exactly what they did at Kansas State a few years ago. They made synthetic wheat lines, and they went through and did that exact same sort of creation of polyploidy from these diploids. First, they had to come up with the hypothesized ancestors, right? So maybe, you know, I'm not sure what the hypothesized ancestor is of wheat or the ancestors, right? Because you're actually looking at 3 different species, but they actually went through, treated, and recreated that in the line. And so this is pretty good evidence that, yeah, that polyploidy explanation for wheat, and probably for a lot of the other, you know, food crops that we looked at, it is common. Have we seen polyploidy formation of species or speciation in our lifetime? In my lifetime, maybe Kevin's lifetime, we have. So when, well, I don't know, do you ever remember, I'm not sure when I-80 was built out to Wendover, but what we see is a lot of polyploidy plant species arising in transition zones. And so when they had to put it, you know, across the salt flats going out to Wendover on I-80, they had to put a whole bunch of, you know, stuff in there to solidify it when they're building the interstate. And so they had to clear away a space, et cetera, et cetera. And in fact, biologists or botanists at the time actually sort of charted and watched the creation of a new polyploid species out on the way to Wendover when they built I-80. And so, yes, you can see that. Why are we calling them separate species? I mean, what's the definition of a species? Those of you, haven't a bunch of you had evolution already? You're in it right now? In the online or face-to-face? So, how do you define species?
Well, are you talking about the biological species?
Good. There's a whole bunch of different concepts, right? So, are you interested in that stuff, Maya? Because somebody's doing a seminar next semester on species definitions. Yeah. So usually if we were to rather than looking at concepts, which really are different criteria that we use in sort of a philosophical sense, what is a species? It's these population of organisms that are sort of on their own evolutionary trajectory. One of the ways that we identify species is if they mate with each other and do they pass genes between individuals. Can individuals in that in that population pass genes between each other, right? And then have offspring that are viable and then continue to pass those genes and mix those genes in. And so with a new polyploid species, we're calling these, sort of these special cases where we have sort of instant speciation. Why? Because are they going to mate back with either of the parental lines? If you're looking at this evolution of the weed, it's going to be a very, very rare occurrence that final, wheat plant is going to be able to mate back with any of those ancestral ones, right? And so they're basically on their own new independent trajectory. Okay, so polyploidy is really important in evolutionary terms. We talked about this before. With polyploidy, you get this massive increase in the genetic material that you have to work with from a natural selection evolution perspective, and so it oftentimes fosters some pretty rapid evolution. Okay, so there are different kinds of polyploidy. So organisms that are polyploidy sometimes arise between hybridization between different species, like we were looking at before. We call that allo. Whenever we see allo, we think of that root word as being what? Allopatric. Who's had ecology? We get to talk about allopatry in ecology. It's other country is basically what it means, and so other species. Polyploidy that arises through the hybridization between different species. Autopolyploidy, same, right, are created by chromosomal duplications within a species. So it's not necessarily, different species hybridizing. But again, that chromosomal doubling is the key sort of event in transitioning from sterile hybrids that are formed between two separate species, whatever mating, and becoming a fertile lineage. Okay, so we also see tissue-specific polyploidy. A very special kind of tissue-specific polyploidy is what we call polyteny that we see in Drosophila that we'll talk about here in a second. There's a process called endomitosis that involves chromosomal doubling, right? So you're basically going through mitosis. That word's escaped me for a second. Mitosis, but you're not going through cytokinesis. So you're going through that nuclear division, but not the cellular division. What's the end result of that? Polyploid tissue, or cells that have lots of nuclei in them, or cells that have lots of copies of chromosomes in them. We see these in different places. In a lot of animals, we don't really see polyploid species, but we do see polyploid tissue from this exact process. In humans, we actually have cells that are polyploid in our liver, in our heart, and our bone marrow. What would be the advantage of this? If it's happening, you might say, okay, what's the advantage? Think about the liver. What's the advantage of having a multinucleated or a whole bunch of copies of a chromosome within a particular chromosomes, or all of the chromosomes within, say, cells that are in the liver? What would you think? Hypothesize. There has to be some advantage of it, right?
Faster regeneration, or is that not?
Faster regeneration of the cell?
Yeah,
or
I'm saying like maybe if like the tissue was damaged, I don't know, like there's
I don't.
chromosomes, maybe
Yeah, that's a good
duplicate
idea.
more.
That's a good idea. I haven't heard of that, but that's a good idea. What else? Faster what? What? But you're on the right track. Faster what? Functioning. If you need a whole bunch of enzymes, right, and you're in the liver to break down whatever or produce something, right? Filtering the blood, breaking down metabolites, whatever, right? And you have a whole bunch of copies of the genes, and you want to turn them on. You turn on all of them, and you get a whole bunch of gene products, and that's probably why we see the polyploidy in certain tissue and not in other tissues. If the sister chromatid, so say you go through mitosis, right? You go through your doubling of your DNA, you form sister chromatids, but then the sister chromatids don't separate out during anaphase. You get what results in what we call polytene, a polytene chromosome, polytene. Here, these are polytene chromosomes in the salivary glands of Drosophila. So if you go through one division, right, but you're not actually separating them out, so you're basically, maybe I'll draw it up here. I think it helps. So let's say, you go through, you start out like this, you go through a doubling, right, of your sister chromatids. Here are your separate sister chromatids, right? But you double without duplicating. How many copies of a gene do you now have? Well, let's look at both of the chromosomes, right? So here are both of the chromosomes. You go through it. You still technically have two chromosomes, right? Here's one chromosome. Here's another chromosome. But now, how many copies of a gene do you have? Four. Or we'll even say there's four chromosomes. But they're still connected here. Okay. So you go through one round of that without the separating out of the sister chromatids. You now have four. If you also now go through another round of DNA duplication without separating out the sister chromatids, now what do you go to? How many copies are you going to have? Yeah, so we're going to go here, 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 here. I'm just drawing this up here. You now have 8 after 2 rounds. If you go through, and this is just an example, but this is a kind of a real example with the salivary glands in Drosophila, you go through 9 rounds. How many copies of each gene or chromosomes are you going to have? There's a formula, right? 2 raised to the what? 9th power. That equals 256, 512 copies. And so that's what you see in the salivary glands of flies. Here, we now, because as you can imagine, when you're doing all of these things, your central mirror then becomes a much more sort of robust structure. We call that the chromocenter. We also see an interesting thing happening in the salivary glands. You see homologous somatic chromosomes, right? So these are somatic cells, but they actually pair with each other. So all of these, so here are your different copies of it. They also kind of come into close contact with each other. So you have these bunch of chromosomes. You have all massive number of copies, and you also have the 2 homologous chromosomes pairing together. With that, if you go and stain those chromosomes, you're going to be able to create really, really high resolution maps because you have so many different copies. So for instance, these are the genes, right? But let's say these gene regions also are uptaking the stain and at the same rate. You're going to have 512 of these. You're going to be able to create a really, really high resolution fine detail map when you stain these chromosomes, and that's exactly what we do with the salivary, the glands or the cells in the salivary glands of Drosophila. And so this is what they look at. Bridges, you remember Bridges in Morgan's lab, the postdoc or grad student, whatever he was. He then did that and created these very, very high detail, high resolution chromosomal maps for Drosophila. This was important because we needed to have a good description of the geography of these chromosomes when we started looking at mutations, because if all of a sudden some sort of mutation arose, you could go to the salivary glands, extract the cells, stain them, and you could look for things like deletions happening or duplications happening and say, oh, okay, we see that there's, in all the flies that have this particular mutation, we see that there's a duplication of this D12 region or something like that. That would then allow you to identify the gene that was coding for whatever, suite of possible characteristics you'd be looking at with those mutations. And so that's exactly what they did. Oh, I thought that was the next slide. That's exactly what they did, and we'll show you here in the next section some of the mutations that they actually located these things happening. Okay. Just a aneuploidy. So aneuploidy is an underrepresentation of a chromosome or chromosomal segment, right? And so, more technically, rather, in the polyploidy situation, the aneuploidy they had different number of chromosomes, but like I mentioned before, more so when we talk about aneuploidy, we're talking about certain chunks, right? And overrepresentation, underrepresentation of sort of genes. So think more along the lines of genes with aneuploidy. Okay, that imbalance oftentimes affects the phenotype. Okay, so here's just some vocabulary words, some sort of new vocabulary words that I want you to learn. Aneuploidy, numerical change in part of the genome. That's that imbalance. Trisomy is a triplication of one chromosome. We've talked about that when we talked about Down syndrome. Hypoploidy is when a segment is underrepresented. Hypo, under, hyper, over, when a segment is overrepresented, right? And then monosomy is the absence of one chromosome in an otherwise diploid individual. So Turner syndrome that we've talked about before would be which one of these up here? Which one of these terms? What's that? Monosomy, right? Trisomy would be Down syndrome. Of these, Turner syndrome would be this one, right? The hypoploid, because you don't have part of it, it's underrepresented. Down syndrome would be a hyperploid because you have it overrepresented. Does that make sense? Aneuploidy would be any of these because you have that imbalance. That aneuploidy suggests imbalance. Right? Are those terms? You have those terms straight? Okay.
Is aneuploidy a type of polyploidy? Like a specific type?
No, not really. I think of these things as separate. So think of polyploidy as when you're doing whole sets. Aneuploidy is when you think of it as imbalance. So if you had to associate one word with a polyploidy, entire sets of additional haploid number of chromosomes. Aneuploidy, just think of imbalance because a certain part of the genome is either underrepresented or overrepresented. Make sense?
Yep.
Okay. Good. So we have a lot of research. Some of the first research that was done in aneuploidy occurred in a plant, Datura, from the genus Datura. And again, good botany stuff. That a couple of researchers, Blakesley and Balling, looked at chromosomal abnormalities in this Datura plant. It's known as Jimson weed. We actually have it. It's a native plant here to southern Utah. They have these nice, has anybody ever seen these before down in southern Utah? If you're out hiking, and especially when there's a nice moon, we call these moonflowers. They open up, they're really nice, white, they look almost sort of succulent. It's surprising a little bit because you're down in the desert and you have these big, leafy plants like this. I used to have a bunch of the capsules in there. I had them around, I don't know what happened to them. But you see them down in the desert, and the first thing that you think about is like, how are these things surviving in the desert, right? These big leafy things. You think there's not a lot of vegetation in the desert, but these things seem to be doing fine without cows or whatever coming and eating them. One of the reasons is that Jimson weed is pretty toxic. So it has a lot of alkaloids. When I was an undergrad, one of my chemistry professors did research. He was a chemist, did research on this. He said it's like tons of alkaloids, basically has stuff that's similar to cocaine and all sorts of hallucinogenics and everything else, right? It's one of the reasons why. Is anybody in here from southern Utah? Nobody? I'm the only southern Utah person. One of the reasons why during my high school and junior college days in southern Utah, I knew 5 people that made tea out of the Jimson weed, right? So the American Indians have this sort of ritual that they go through, and they drink the tea, and they go on like these vision quests and find their animal spirit and everything. And so obviously, teenagers think they can do the same thing. And so they get the roots of these Jimson weed, cut them up, throw them in an old sock, make a big barrel of tea from it, and then drink it. All 5 individuals that I knew, still know some of them, ended up hallucinating for anywhere between 24 and 48 hours solid without sleeping, and all 5 of them ended up in the hospital, in the emergency room. So this is really, really toxic stuff. And so, what's that? Hey, I want no part of this. No tapping me for information. No, I don't. So no, I don't know how much tea they drank, but it was too much. Even a little bit is probably too much Jimson weed. So that does a good job. So a cow that maybe nibbles on a little bit of this is going to be whacked out for a long time and never ever going to eat it again. So it's maybe a commentary on humankind, that we're the only animal that happens to, and so then we continue to go back and cultivate it, with lots of other things. But it's usually defense mechanisms. With cannabis and anything else, the reason that we feel these highs and things like that, mushrooms, it's a defense mechanisms by these plant and fungi that usually works, except when humans come around and then use it it's a motivator. Okay, so anyway, that's sort of a side story. But Jimson weed, they have these capsules where they keep the seeds. When these researchers were looking at the Jimson weed, they noticed that there was a lot of variation in the capsule morphology when they went through and looked at and scored the chromosomes for each of these variants in capsule morphology. What they found was the diploid Jimson weed, or Datura, looked like this. This is what the seed capsule looked like. When they had ones that looked like this, all these different variations, they would go through, look at them, and they would find that they were triploid at these different chromosomes. So there's 12 different chromosomes. Say, for instance, when they were triploid E11, the capsule would look like this. When it was triploid E8, the capsule would look like this. Triploid E4, the capsule would look like this. Again, compare this to human triploids, or trisomy. When we have trisomy in humans, this plant can accommodate the trisomy in every single one of its chromosomes with, not much difference. These were fertile, et cetera, et cetera. You just had these morphological differences. So they're much better at accommodating these triploid situations. Question?
I just wondering, so with humans, trisomy causes Down syndrome. In this case, it seems it's beneficial.
Yeah, so in this case, not only are they being able to accommodate it more, but now you have more genetic variation. So if for some reason a capsule, there's an environment that a capsule that is all robust and big like number 11, you would maybe then use that for some sort of advantageous selective advantage. In plants? I think more so a trisomy than a monosomy, because with a trisomy you have added genetic, you have a whole other suite of genes, and really when you have these extra DNA, what that does is it sort of decouples you from having to keep whatever the genes are on your original one to produce the function that they're producing. Now you have an extra copy, and those things are free to sort of tinker with. So evolution can then tinker with that, and we see that in blood types and stuff like that, even in humans, where all of a sudden you can have a few mutations and whatever, and then all of a sudden they're able to sort of have generate a new sort of function, and you have that freedom to do that because you already have your your other genes here providing the physiological function that they need to provide, and you have this extra stuff that you can sort of tinker with and mess around with. Kevin, did you ever?
That's a question.
Same thing.
If they were variation or added to pool variation.
Yeah, and that's exactly the benefit of those sort of things. So not only can they can accommodate it more, but they also can use it for beneficial things. And I don't know the exact story with Datura, if some of these capsules are more advantageous than others in certain environments, or what the history is behind them. But sort of the take-home message is, that plants can do this, that we do see these polyploidy situations or aneuploidy situations. Okay, so let's go back to humans. So we've talked about trisomy, we've talked about Down syndrome in humans. We're just going to go through a couple of cases of trisomy quickly. In humans, here's trisomy 21. We've talked about, here's the karyotype of a human. Again, trisomy, the 21 chromosome 21, probably doesn't have a lot of super essential genes, so it's able to accommodate an extra, or for some reason we're able to accommodate that much better than any of the others. We'll talk about a couple of other trisomy situations that aren't as common. For sure, Down syndrome is by far the most common trisomy situation, so that tells us that probably for some reason we can accommodate that extra set. Maybe there's fewer genes here that we don't have to deal with the extra production of the gene products. Maybe the genes that are on there, the composition aren't as essential as some of the other chromosomes, because you might say, okay, well, if chromosome 22 is smaller than chromosome 21, why don't we have trisomy 21? It probably has to do with exactly which kind of genes are on chromosome 22 that messes it up when we have a trisomy. What are just some of the the suite of characters that are associated with Down syndrome? And there's a lot of variation in this. We see this, we've talked about that environmental component. With Down syndrome, there's lots and lots of variation, but these are just some of the different characteristics that are associated with it: short stature, loose joints, broad skull, etc., large tongue, stubby hands, those sort of things. In the palm, this is the old term, not so PC term, the simian crease. If you look here in the hand, it goes straight across. You also have a particular morphology of the tongue. How does the trisomy occur? Specifically, how does it occur in humans? It's usually occurring. It's usually occurring with non-disjunction, but it can occur, not how, but where does it occur? It can occur in meiosis 1 or meiosis 2. Christian, question?
Can they tell things pregnant if they're going to have Down syndrome?
Yeah, we'll talk about that here in a second. How do we test for these chromosomal abnormalities during the gestation period? Yeah, there's two general techniques. So if disjunction, we know that trisomy is a result of disjunction. We can say, okay, the disjunction can either occur in meiosis 1 or it can occur in meiosis 2. In this case over here on your left, it's occurring in meiosis 1 here, so both of these, we'll just say chromosome 21, go to one side, none of the chromosomes are on the other side. You then have gametes that form from this. This is going to be missing a chromosome 21. These gametes are going to have an extra chromosome 21. If they get fertilized by a normal gamete, you're then going to have trisomy 21. So this would result in Down syndrome. This would result in Down syndrome. What would this result in? Yeah, probably a spontaneously aborted embryo. Okay, so in meiosis 2, so say you go through meiosis 1 in a normal sense, right? And then non-disjunction happens here in meiosis 2. What you see is that here you have that overrepresentation, underrepresentation. This would be Down syndrome, this wouldn't. But this one can go through the normal division still, right? And so if you had to choose between one or the other, you'd probably choose meiosis 2 to have the non-disjunction, because you're still going to have some gametes here that are normal gametes, whereas here, if the non-disjunction happens in meiosis 1, you're not going to have any gametes that are in very good shape. Here, this is a table of a bunch of aneuploidies that result from non-disjunction in humans. You have Down syndromes, you have Patau syndromes, Edwards syndromes, Turner's we've talked about, Klinefelter's we've talked about, Downs we've talked about. How many of you have heard of Patau or Edwards syndrome before? Just go through, read these. They do actually, like Down syndrome, we have individuals with these syndromes. Here are just some of the phenotypes. Just in general, if we look at Patau's syndrome, here, that's trisomy 13. A lot of times that's associated with cleft palates. So you kind of think, Patau syndrome, cleft palates. Edwards syndromes, they get this sort of Elvian phenotype with ears that are sort of set lower down on the head that kind of gives them that elfish kind of appearance. That's Edwards syndrome. Turner syndrome, we talked about before. Turner syndrome, often a phenotype that's associated with Turner syndrome is kind of this webbed neck here. You sort of have this fold of skin that causes the folks with Turner syndrome to sort of have that L neck, that web neck. But again, with these, just like with Down syndrome, there's a big variation in these phenotypes, and a lot of times that variation has to do with the background genotypes, the background genome, what the other genes are coding for, and it also has to do with environmental, different environmental things that. And so it's important that we, if you were having an offspring with one of these conditions, that you know as early as possible, probably, that it's happening, because then you can take, measures that would maybe lessen these different characteristics of the phenotype. Okay, so Turner syndrome we also know. So here's the origin of Turner syndrome, right, in the egg and the sperm. But sometimes we actually have tissue-specific Turner syndrome. In this tissue-specific Turner syndrome, you have a normal zygote, right? As the zygote begins to go through mitosis, different rounds of mitosis, you have an overrepresentation and underrepresentation of certain tissue cells, right? And so then you will see Turner syndrome in particular cells. And so it's not, so we mention this just because, so that you know that even sometimes in the tissue, you can have over and underrepresentation of chromosomes. Okay, so back to that earlier question from Christian, how do we detect these chromosomal abnormalities? There's basically two kinds of tests that we do. There's more than that, but we're just going to talk about two. One of them that maybe most of you have probably already heard of is amniocentesis. Amniocentesis is just basically sampling the amniotic fluid. The amniotic fluid, has anybody ever seen amniotic fluid before? Nobody? It's not this, nice, clear Caribbean water, right? It's full of all sorts of muck, because it's in there for a long time. There's sloughed off cells hanging out in there, et cetera, et cetera. Fortunately for us, in that amniotic fluid, because of the sloughed off cells from the embryo, we can actually go in there, sample that fluid with that fluid, centrifuges, separate out the cells, grow them up, and expose them to a whole bunch of different biochemical analysis. But then also we can go in and count the chromosomes, right? So you go in there, you count the chromosomes, you line them up, you make a karyotype. Here you go. If for some reason a pregnant female wants to get an amniocentesis, for whatever reasons, they're going to go through and do this whole suite of tests, and that's part of it, looking at the chromosomal number. And then they could identify in utero an individual that has trisomy 21. This is one technique. There's another technique that you can do earlier on in the pregnancy. So this is usually later on in the pregnancy. I don't know when the earliest you can do an amniocentesis. Has anybody ever had an amniocentesis? Anybody know anybody that has an amniocentesis? No? Maya, what was your question? Put it in, yeah. I mean, yeah. Oh, through the uterus? No, I think it's coming in from the side, but I actually don't know for sure. Do you know? It comes right in through the stomach. Yeah. Yeah. So the other earlier detecting there. So this is sort of the less risky, right? Later detection that you can do this in analysis. There's an earlier one called chorionic villus biopsy, where you actually sample part of the chorion, right? This happens at 10 to 11 weeks, so really, really early on in the development of the embryo, and they just kind of go in there and take out a little bit of it, right? Here's that chorionic sac, and they just kind of go and take out. The reason why they only do this when there's a lot of evidence that there could be complications, where there's a history of lots of complications in the family of either of the parents, because when they do this, this really increases the chance of spontaneous abortion, and so they only do it under certain situations. But it's pretty early on. This is 10 to 11 weeks into the pregnancy, so it's fairly early on. But these are ways that they test for those chromosomal abnormalities. There is a premium to that, like we were talking about, because if you do know early on, you can do things to make it so that the eventual phenotype is lessened, the dramatic effects of the phenotypes are lessened, things like that. And then maybe even in certain cases with chorionic villus biopsy, maybe the couple would, the mother would opt to abort. You just never know. Okay, so deletions, duplications of chromosomal segments. So now we're kind of going down to the smaller. We started at polyploidy, whole sets. So look at aneuploidy, this sort of on a smaller scale. Now we're going to look at just parts of a chromosome. So you can have either a deletion or a deficiency of part of a chromosome segment, or you can have a duplication of part of a chromosome segment. This probably, based on what we've learned already, how do you think you would get a deletion of part of a chromosome? Or how do you think you'd get a duplication of part of a chromosome? How could that occur? Is there going to be a little chunk that just gets placed in there? You have the knowledge. You just have to think about it. What sort of process? Think back to meiosis. So somebody's going to say something. So recombination, or who said that? Who's first say crossing over? Somebody, I heard it somewhere. So crossing over, yeah. So if you get unequal crossing over, then, most of the time you're supposed to, one arm of a sister chromatid and the other arm of a sister chromatid touch, get all cozy at the chiasma, right? And then they exchange parts. But sometimes they're not going to exchange equal amounts of parts. If you get kind of not as much as you gave, then you're going to have a deletion or a deficiency of part of your chromosome. If you received more than you gave, then you're going to have a duplication. And again, this could have consequences. This could have evolutionary consequences, because in some case now you may have an extra copy of a particular gene or a particular group of genes. Okay, so we do have human conditions where we do have deletions in the small arm of the fifth chromosome, number 5. Remember this nomenclature: chromosome number arm petite or small arm at band 14. We have a deletion when we call this the Cry-du-Chat syndrome, or the cry of the cat in French syndrome, because individuals with this may make this kind of meowing sounds, and it is a result of a deletion of part of the number 5 chromosome. If we look at those polytene chromosomes in Drosophila, we can actually get a really good sense. We can get, like I was mentioning before, we can get a really good sense of where this stuff is happening because we have such a high resolution map. If you were to look at a polytene chromosome here, and you're looking at it under the microscope and you see a big bulge right there, what do you think that would be? That'd be a duplication or a deletion. It just depends, right? Because if you have a deletion of part of it, that's not going to have anything to bind with. Same thing with the duplication. But the point is that you would recognize some sort of chromosomal abnormality. If you had a duplication, how would you detect that? Remember, it's polytene chromosomes. How would you detect either, really, but especially a duplication? You would look at the banding pattern. If the banding pattern was here, here, and then all of a sudden this bulge came out with that same banding pattern, you say, okay, this is a duplication. if this banding pattern you didn't see anywhere around there, you say, okay, it's probably a deletion on the one side and then a duplication on the other. And so the polytene chromosomes really allowed us to sort of anchor down and figure out exactly where, and we'll talk about this in chapter 7, exactly where these mutations were occurring. And that's really okay. So here in, just as a real life example, with the bar mutation that we talked about before, here's a wild type. This is what the normal chromosome looks like. When you have the single bar mutation, what you see if you look at these banding patterns is a duplication of that same banding pattern, a single duplication here. If you have a double duplication, this is the phenotype that you get. So you go from the double bar, which is this really tiny eye, sort of this intermediate eye, and you associate that with this particular region. In this particular region, then you're saying, okay, this region has something to do with eye morphology. So it's the first step in really identifying where these genes are and what they're doing. And then here's the wild type. Okay, so there's some real life examples of that. All right, so that we're done with that section. Pretty good time. That's good. I don't think we're going to take a break today. And I'm probably more disappointed than anybody else. But I think we just have to keep cranking away. Okay, so we've talked about cytological techniques, polyploidy, aneuploidy. Now let's just talk about rearrangements in chromosomal structures. So here we're not necessarily having an overrepresentation or an underrepresentation. We're just getting a rearrangement of how the genes are housed in these chromosomes. And again, like with all the rest of these have important evolutionary consequences that we'll talk about in a second. Okay, so a chromosome may become rearranged internally, or they may break off and link up with another chromosome. And so there's a whole bunch of, just sort of looking at everything that we've talked about today, thinking about, back when we talked about meiosis and the crossing over, chromosomes and chromosomal structures are really much more sort of a dynamic thing than we ever thought originally, right? They're crossing over, they're exchanging parts are breaking off and attaching to other parts. There's a lot that can go on with those chromosomes. They're not sort of this, set in stone structure. Okay, inversions, and this is the one that we'll mostly talk about. We'll just define it here. Inversions are when segments detach, they flip around, and then they reattach. And so here you have a normal chromosome, right? Here, these letters are just representing your run-of-the-mill genes, right? So here you have gene A over here, near the telomere. You have gene F down here by the centromere. You have gene G here, down kind of by the centromere, and then gene H again out towards the telomere. What kind of centric chromosome is this, based on our nomenclature? Metacentric? Pericentric? Acrocentric? Did we talk about this? Meta, what does meta mean? Middle. Is the centromere in the middle? No. So this is, we'll say this is, if I said, is this a metacentric one? No, it's not. Okay. So what happens is you get a chunk of this chromosome, detaches, flips around, and then reinserts itself, right? That happens. That's an inversion. And so basically we can detect inversions by looking at the gene sequences and by looking at the gene orders. So maybe we have some of those probes like we were talking about before from, given DNA. We attach these probes. They're able to light up where these different genes are, right? We go through. We see it detach, flip, reinsert. And what we find is now the gene order is completely different, okay, on one of the chromosomes. That's an inversion, and we see this happening frequently within the genome. If that were to happen, so here is, if that were to happen and that inversion included the centromere here, we would call that a pericentric inversion. It includes the centromere. And so here now, if that inversion happened and included the centromere, you can see the gross morphology even, without even being able to look at the gene order, right? You say, oh, okay, something's going on. These two are the seems like the centromere is, the centromeres don't completely line up. Okay, that is the result of a pericentric inversion. A paracentric inversion is, just say some other little part that doesn't include the centromere. It breaks apart, flips around, and reinserts. That's what happens with paracentric inversions. Okay. So these inversions. Given that, when these inversions happen and chromosomes go to line up in meiosis, when they pair with each other, if you pair an inverted chromosome with a normal chromosome, the different parts, the regions with the different genes will tend to try to link with each other, right? And so in a normal case, let's just say we had two normal ones, A would pair with A, B, that's kind of how the chromosomes would be oriented. They still try to do that after an inversion. And so here on this one, I'll sort of show you. On top here is the inverted chromosome. Down here is the normal. Below is the normal chromosome. And in order to still get those same regions lining up with each other, you have to kind of stretch it. So this chromosome, and you might not be able to see this in the back, it's kind of like a roller coaster, a loop on a roller coaster. It comes in here, flips around like this, and goes through here. So you get this big stretching between this region of those chromosomes. So they're still able to line up during meiosis. The result of this, or what happens with this, if you were to have crossing over occur, let's say crossing over occurs, well, I think I have it on the next slide, in between E and D is, I think. Yeah, if crossing over occurs between E and D, what would the gametes look like? And so here you have this chromosome breaking here, this chromosome breaking here, and switching those parts. Like break it here, attach it here, break it here, attach it to there. Let's just look at the one. If you follow that through, you're going to have A, B, E. Crossing over occurs here, D, C, B, A. What is that gamete going to look like? It's going to have an overrepresentation of some genes, and even more importantly, it's not going to have some of those genes. And so when you have crossing over occur, it really jacks up the gametes. What then could there be an advantage of that? What then would that do to those genes in the inverted region? I mean, are you likely going to have, so say you have that so let's just do this one. Maybe I should draw it here. Let's just do this one. The gametes that would result from a crossing over that occurs is going to be, what did we say this one was? A, B, E, right? A, B, E, and then A, B, E, and then D, C, right? Did you ever see what I'm doing? A, B, E, D, C, B, A. D, C, B, A. Okay, here's that gamete. Sorry, this isn't A. The other one would then be what? H, G, F, C, D. Ever see that? H. Well, I'll do this other way. It would be H. Here, we go there. H. We're going to do it over here. It's too hard to look. Yeah, the second one, that's important. The first one, what doesn't that one have? Well, let's just talk about this one before we do the second one. Well, maybe we don't have to do the second. So what is this? What's missing on here? The centromere. Do chromosomes need centromeres? Probably, right? What else is missing here? FGH. So FGH, so you're missing three genes and whatever other genes are in that area that maybe you don't even know about. You're missing a chromosome, right? You have what? 2 copies of A, so now you're getting this imbalance. 2 copies of B, right? So overrepresentation of some, underrepresentation of some, and no centromere. So what's going to happen? So what's the result of this? You're basically screwed, right? You're not going to fertilize. You're not going to form anything, right? The other one's going to have same thing: underrepresentation, overrepresentation, and 2 centromeres. Is that going to mesh up in meiosis? Yeah. So this isn't a good situation. So why do we see it? Why hasn't natural selection come in and why do we see inversions pretty frequently in all organisms' genomes? There has to be an advantage. If this is a massive disadvantage, what does it do? What does it do to crossing over between those areas? If it does cross over, what happens? Yeah, nothing's going to result. You're not going to pass that on. So in essence, what it does is it restricts crossing over. It locks that region of genes in a certain combination of alleles of those genes together and doesn't allow crossing over to occur. Well, crossing over occurs, but nothing that results from it. So only gametes that form when there's no crossing over between these in this inverted region are going to be produced for reproduction. So essentially what that does is it freezes those combination of alleles. It locks them up and inherits them kind of as a single gene. Is that important? What's the whole purpose of crossing over? When we were talking about it before, what does it do? It mixes up, it creates genetic variation, right? It mixes up those gene combinations, right? If you're not crossing over, what are you doing? You're locking in that specific gene combination or allele combination of those genes. But what if it's a good one? What if it's one you don't want crossing? You don't want you don't want to mess it up. This is you've struck upon, the perfect sort of combination, as we see with asexual organisms, right? And then you say, oh, I don't want that. That's going to be beneficial. That's what happens. So inversions actually can be beneficial. We see that too, and this is, we'll just go through this sort of natural experiment really quickly, and we see the adaptiveness of inversions even in the natural world, when we have combinations of alleles that work well in different environments. If it works well in a certain environment, natural selection will favor those inversions because it's locking up that combination. It's a great combination. Let's keep it. What we see in Drosophila subobscura is that when we look at their genomes, we see that they are polymorphic, meaning they have some chromosomes that have inversions in 5 of 6 chromosomes, right? So there's inversions. There's a lot of inversions. 5 of the 6 chromosomes have some inversions. That doesn't mean that all of the chromosomes have those inversions. Maybe either one of the two have those inversions, but there's a whole bunch of inversions. Drosophila subobscura is native to Europe, North Africa, and the Middle East. And what we find is that frequency of those inversions will vary with latitude. So the subobscura that are up here in Europe, right, they have lots and lots of inversions. The subobscura that are in the Middle East and in Africa, North Africa, they don't have very many inversions. So researchers begin to think, wait a second, okay, with all of these inversions, maybe having all those inversions are locking up these genes that are favorable for that environment. What is, if you look at, even if you go from sort of Northern Europe to Mediterranean Europe, down into the Levant or the Middle East, and then down into North Africa, what's the environmental transition that you see in that? Which of those areas is more moist and wet? Which of them is more dry? You basically have this gradation from what to what, starting in Northern Europe. Wet to sort of Mediterranean, chaparral type stuff, to dry and desert. That same trend you see in the frequencies of the inversions. In the wet environment, you have a whole bunch of inversions. In the dry environment, you don't have very many inversions. So researchers thought, well, that's probably because those inversions are locking up a suite of alleles of these genes that are favorable for that wet environment. As they go down into the dry environment, they're not so favorable. Natural selection isn't selecting for them. And if there's no natural selection selecting for that particular combination, there's no benefit of it. Is there a downside to having those inversions? Yeah, you don't produce, you have all those jacked up gametes, right? And so once that balance is reached where it's not favorable, you're out of that environment, the flies that don't have those inversions then can reproduce better than the flies that do, and so you lose those inversions. And so it's really interesting. So that's a really cool sort of system that shows those inversions in a real life situation. So different combinations form what we call these clines, which are sort of like topo map, gradations of frequencies. But what's even more cool about this system is we see this pattern, we see this parallel pattern, there's this correlation, but is that causation? No, I mean, not necessarily. So it's like this cool pattern, but in order to really nail down that these inversions are in fact advantageous and that they're changing and that they're probably locking up these genes that are well suited for wet environments, it'd be nice to do an experiment with it. And in fact, in this case, we have a natural experiment that went on where you had Drosophila subobscura showing up in the 70s in two different places outside of the Eastern Hemisphere. You had them showing up in Port Townsend, Washington, which is kind of up here just across the bay from Seattle, and north a little bit. Again, what kind of environment is that? A wet environment. And then you had them showing up here, down here in Puerto Montt, which is right down here. It's a port town that I actually lived in for a little while. In Chile, right at the sort of southern part. This is sort of Chilean Patagonia, really wet environment, rain 10 months out of the year. So really wet environment, really similar to the environment here. You had Drosophila subobscura showing up there. The flies that did well in these places, in these wet environments, had a bunch of inversions. What happened after that? The flies began to migrate. So here the flies begin to migrate a little bit north. Again, more wet environment. And then south, if you go south of Seattle down through Oregon and then Northern California and then down into like San Diego. What's San Diego like? It's nice, right? But it's dry, right? Then in Baja California, even more dry. So you're going from wet to dry. You have a natural experiment of this fly going down. You have a natural experiment, sort of the opposite here in Chile. After you go down into Patagonia and the Straits of Magellan and stuff, really wet, lots of rain. You go up north through Santiago. Santiago's kind of like California, Chaparral, kind of Mediterranean. Viña del Mar, all the way up to Antofagasta up here, the Atacama Desert, again really dry. So perfect natural experiment going on. We'll stop there, and I want you to think about, for next time, what do you expect to happen to the frequencies of those inversions as those flies migrate in both those directions? Okay, we'll stop there. Any questions? Again, for those of you who came in a little bit later, here's a major announcement. Come up here, read this, take a picture of it, and then talk to me if you have any questions. Questions, Robert? Okay, so what do we see? So here are the results. This is the data that were collected. Here is your South America. Here is your North America, right? On the x-axis, you have the latitude, right? On your y-axis, you have the frequencies of inversions. If you were to draw regression lines, this regression line would look like this, right? This regression line would look like this, right? And so what do you see happening? You see as you here, so North America, right? As you get farther north, what's happening? Are your frequencies increasing or decreasing? Increasing. So they're increasing because it's more wet the farther north you go. Here in South America, the farther south you go, right? South latitude, the increase in south latitude. What's the regression looking like? You have that same, pretty similar slope, right? Maybe a little bit different. But as you get farther south into Patagonia, Chile, what are you seeing? An increase in frequency. So these two natural experiments, does this add information, or does it? Do you? You can test it statistically, right? You can test the slope of that regression line statistically. Is it statistically different from zero? Most likely, just kind of eyeballing it, it's going to be. Therefore, would you reject your null hypothesis if your null hypothesis was that the inversions, the frequencies of inversions are locking up those alleles, or would you fail to reject it? You would not reject it, right? This is added information, this is added data that suggests that in fact these two natural experiments reinforce that idea that inversions can have an evolutionarily beneficial role, and that role likely is that it's locking up those combinations of alleles. And so within your genome, you have, in essence, a chunk of your genome that is reproducing kind of asexually. There's no mixing going on, and then the majority of your genome there, you're reproducing sexually, and you're trying new combinations of alleles, et cetera, et cetera. So in essence, you have the best of both worlds, right? Part of your genome you're locking up in the particular combination, and part of your genome you're still tinkering with and toying with from one generation to the next. Okay, so next thing we're going to talk about are translocations. Translocations are when a segment of one chromosome detaches and then reattaches, and so kind of like crossing over, where you detach and then, in crossing over, you detach and reattach to your homologous chromosome. In this case, you reattach to non-homologous chromosomes. You can have reciprocal translocations when you have two pieces of non-homologous chromosomes are exchanged, and it's reciprocal in the sense that there's no net loss of genetic material. But you also can have non-reciprocal translocations where you actually do have a net loss or gain of chunks. It's not an equal, that's not spelled right, reciprocal. This is reciprocal. Well, I need to change that. Where you have non-equal exchange of genetic material, and this is important again from an evolutionary perspective. Okay, so here, structure and pairing of reciprocal translocation chromosomes, and so here we have, let's just say, we're going to pretend it's a Drosophila. This maybe is chromosome 1, chromosome 2. You have a chunk of this from this sister chromatid, detaches and attaches here. You have a chunk of this non-homologous chromosome that detaches and then reattaches here. And so now, on one of your chromosomes, the majority of it is from chromosome 1. I'm looking at this second one down here. And then a little bit of it is from chromosome 2. And so you have no net loss, so that's a reciprocal translocation. But what does change? I mean, can you see, okay, you still have the same genes. You still have, if there's one allele from, we'll just say this is gene D, you have them there. They still maybe are heterozygous. The only difference is what? Where they're located, right? And so can you think of complications that maybe could arise from this? From having, it's all the same. I mean, your genome's all the same. You just have the net total of all of your DNA is still the same. So you might think, okay, it's not a big deal, but where could it be a big deal? Do you think? You might get alleles and you might get combinations that don't work in essence. But are you changing the combinations here or not? Do you think? All right, Corey's saying no. You're not changing it, right? You're just changing what? The location or the order, right? And so it might, like what Carl's saying, it might have an effect, kind of like inversions, where it's going to maybe have an effect on the crossing over. So again, we come back and there may be some complications in meiosis. What happens in meiosis? In meiosis 1, how do chromosomes line up? With homologous chromosomes. So is this chromosome homologous to this one, or is it homologous to that one? Some say here. Corey says here. Some say here. Maybe it's homologous to both of them, right? And so how is it going to line up in meiosis 1? It's going to be a kind of a lining up. And so here, this is how the chromosomes would line up, all right? So you actually, rather than just having two homologous chromosomes, you have this combination of both of those chromosomes where B is going to line up with B, D maybe is going to line up with D. If, say, your spindle fibers come in and attach here at 1 and here at 3, and pull these chromosomes to the right, attach here at 2 and here at 4, pull those two chromosomes to the left, could you see complications with that? With the gametes that eventually derive from that? What's that, Elizabeth?
You have an imbalance of D and B and
Yeah, so over here on the left you have an imbalance, right? You have two D genes, right? And over here on the right you don't have any D, right? You have 2B and no D. Over there on the left you have no B. Is that going to mess up if you're completely missing a whole chunk of a chromosome and all of the genes that are associated with that chromosome? Yeah, likely going to be messed up. And so here in this next slide, so they go up, you know, centromere 1 and 3, et cetera, just what we said. You're going to get disjunction, right? Okay, obviously there's problems with that. So let's go back. So if that's happening, we know that that happens. How is the cell dealing with it? Can you think of a way that the cell, or a way in meiosis, that this can occur without an imbalance? Let's go back. Let's go to the next slide where we'll go back and look at maybe the spindle fibers somewhere else. Maybe instead of going left to right, they go up and down, right? And so in this one, centromere 1 and 2 go up, centromeres 3 and 4 get attached and go to the same side. Do we have problems here or not? Have we solved our problem? So the cells, the gametes that form from on top of the screen, what do they have? Do they have a full complement of all of the genes? Somebody says yes, somebody says no. What do they have? They have 2 A's and no C's. The ones on the bottom have 2 C's and no A's. So again, are we in bad shape? Yep, we're again in bad shape. We're going to produce gametes that don't have any, certain chunks, and are missing a whole suite of genes, and so most likely they're going to spontaneously abort or never even fertilize to begin with. So, what's our solution? Can you think of it? Think of these things spatially. Is there any way to separate out these chromosomes that would in fact give you a full complement in all of the gametes that are derived from them? You can't go to the side, you can't go up and down. Diagonally, how would you go diagonally? Maybe you have, so let's think of this in like 3D, right? Maybe, I think this is where Chris is going. You're redeeming yourself from that wrong answer before. Okay, maybe the centromere or the spindle fibers are coming here, attaching to here and here, and maybe that pulls it out towards you, right? And maybe this one and this one pulls it in towards the chalkboard. If you were to have that sort of three-dimensional pulling, what you have, and this is what it has, centromere 2 and 3 go to one pole, centromere 1 and 4 go to another pole. Are we okay now? Yes, we have gene A, B, C, and D on the lower right. On the upper left, we have C, B, A, D, on the upper left or out towards the class, and the other one in towards the chalkboard. And so you have to think about these things as a cell from this three-dimensional perspective. You do, in fact, if in fact that's how they end up migrating to the different poles of the cell, you would have a full complement. So this is, it's kind of like the inversions where you have a bunch of cases where you're kind of screwed up, but a few cases where it's okay. And so it's similar to that. Okay, so we have that. So one of the sort of take-home messages for this whole section, for this whole PowerPoint, is that there's a lot of stuff going on in meiosis, right? Usually, in zoology 1110 or whatever other general biology class you had, you sort of think, okay, here are these chromosomes, they line up, and then they pull one side to the next, and we sort of gloss over what's really happening during the lining up in meiosis. And what is happening is there's a lot of things going on. It's very dynamic. It seems kind of weird to us that we have chunks of chromosomes breaking off, reattaching, et cetera, but there's a lot of stuff going on that if it doesn't go right, it can screw up the eventual gametes, right? But given the fact that we see that there are times that it is okay, that tells us how much positive selective pressure there is for these exchanges, right? And what then is the benefit of these exchanges? It has to be a pretty good benefit. What is that pretty good benefit? Why, if there wasn't a benefit, you wouldn't have it, because there's obviously a cost, right? What's the plus? What's the positive, Elizabeth? Variation. Good, yeah. So variation, you're switching up you're tinkering, you're switching up those combinations, And by doing that, you're able to adapt and change and evolve to new environments. Therefore, there's a pretty substantial positive effect of that, even with the cost of producing a whole bunch of jacked up gametes. Okay, so compound chromosomes. Let's just talk about these for a second. They're formed by the fusion of homologous chromosomes, or sister chromatids, or homologous chromosome segments. So here is sort of the fusion of chromosomes, and this can happen a bunch. There's a section in the book that talks about Lillian Morgan, where she crossed homologous recessive X-linked females with wild-type males, and so again, the formation of these compound chromosomes helped us to understand mutation, helped us to understand how things are inherited. What she used compound chromosomes to explain, kind of like when we're looking at Albright, is it Albright? I can't remember, the grad student, Morgan's grad student, who was looking at non-disjunction to explain deviations in the pattern. Sort of Lillian Morgan is doing the same here. Basically, and we'll go over this pretty quickly. I just want you to look at it in the book. She explained, how the patterns of the wild-type males looking at these compound chromosomes to explain that. So I know I didn't do a very good job explaining that. Just go over that, just kind of read and look at that. Okay, so the last part that I did want to show you, though, and I do want to spend a little bit more time on, are Robertsian translocations. And so in Robertsian translocations, you have a fusion of two non-homologous chromosomes at their centromeres. And so then this is important. So you have not only the breaking up, or you have fissions that we talked about. Remember when we talked about Scoloporus grammicus and all the variation in the karyotypes of those, they were the mesquite lizards, where you have a whole bunch of what are called Robertsian fissions going on, and chromosomes splitting up. You also have chromosomes coming back together. So again, there's just all this crazy sort of dynamic stuff going on with the chromosomes. Sometimes you have the fusion of two chromosomes. And so this is showing you two acrocentric chromosomes fusing at the centromere. You lose a little bit of both of those chromosomes. They're at the end. What do we generally find at the end of chromosomes? Pat? Yeah, telomeres, right? What are telomeres made up of? What kind of DNA? We call them micro and macro satellites. They're repetitive DNA. So it's like maybe 1,000 copies of AGCT, 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 et cetera, et cetera. So they're usually not coding. So the fact that you're losing that little chunk isn't a big deal. And then you form a big metacentric chromosome from two acrocentric chromosomes. And so this also happens. Is there a benefit? Is there a plus in this? Hard to say what it would be necessarily, but is there evidence that this in fact does happen? And yes, there is, and so I think this is the last slide. We're going to end this PowerPoint by looking for evidence of Robertsian translocations in human chromosome number 2. What kind of evidence would you look for? If you're looking at the DNA, what would suggest that there is in fact this does in fact happen? You'd want to look for genomes, right, that differ in, say, 1 chromosome number, or 2 if you're looking at the diploid state, right? And then you'd want to look at you could look at these banding patterns, right? So if you have this fusion, you should get similar banding patterns, not only similar banding patterns, but you could do it on a higher resolution scale, right? You could actually look at the sequence, and you should see sequence of, sort of telomere or centromere type sequences within here. If it was 2 acrocentric, if it was 2 metacentric chromosomes, you'd get those would generate different hypotheses. And so here, some of you probably have seen this already. It's just a little chunk of one of your labs. I think it's 1110 lab, where you watch the whole thing on Dover. Did everybody see the Dover Intelligent Design on Trial? What's that? Yeah, it was playing the other day. Yeah,
It was
exactly.
playing, like, all day over here because I was studying in the room.
Yeah, okay. So here we're going to watch just a couple minutes of it. We'll then take a break. We'll then take a break, and then we'll come back and get into chapter 7. Okay.
The two scientists discovered the role DNA plays in heredity. They founded a new science called genetics that put Darwin's theory to the test. The cells of all great apes, like chimpanzees, gorillas, and orangutans, contain 24 pairs of chromosomes. If humans share a common ancestor with apes, you'd expect us to have the same number. But surprisingly, human cells contain only 23 pairs.
Now, we share a common ancestor, but what happened? Is it possible that in the line of evolution that led to us, a pair of chromosomes from a primate ancestor just got lost, just got discarded? Well, the answer to that is no, it's not possible. The reason is because every primate chromosome has so many important genes on it that the loss of both members of a pair would be fatal. You wouldn't even get through embryonic development. So there's only one possible explanation. And that explanation is that in the line that led to us, two chromosomes that were separate in other primates became fused to form a single chromosome. And this is why evolution is a science. That possibility is testable. We want to be able to look at our genes and find that we carry a chromosome with the marks of that fusion on it. Now, how would we find that? It's easier than you might think.
Typically, on the ends of every chromosome, you should find special genetic markers, or sequences of DNA, called telomeres. And in their middles, you should find different genetic markers called centromeres. But if a mutation occurred in the past, causing two pairs of chromosomes to fuse, we should find evidence in those genetic markers. Telomeres not only at the ends of the new chromosome, but also at their middles, and not one, but two centromeres. Finding a structure like this in our chromosomes would explain why humans have one pair fewer than the great apes.
If we don't find it, then the case for common ancestor for our species might be refuted. But if we do find it, it would be powerful evidence in favor of evolution. So all we have to do is to scan the human genome and see, do we have a chromosome that has these marks, telomere DNA in the center and two centromeres? Well, the answer turns out to be we do. And it's human chromosome number 2. The evidence is unmistakable. We've suspected this for a very long time, and in 2005, a definitive study was published showing that chromosome 2 has the exact fusion point, almost the point at which the Scotch tape holds those two chromosomes together perfectly. The closer we look at our own DNA, the more detailed a glimpse we get of our own genome, the more powerful the evidence for our own evolution from common ancestor with other species.
Okay, so let's take a break, and then we'll start on chapter 7 when we get back. That's a good example of, pretty famous, right? Robertson translocation. Or Robertson fusion, sorry.